Hello and welcome. In this video, I'll be talking about comparing sequences, and in subsequent videos, we'll be talking about dot plots, scoring matrices, and dynamic programming. First, let's do some review. What is the appropriate way to make an iterator I that with every iteration becomes each value in the array? So if we think about this, we're asking I to become each value in the array. So that's distinct from asking it to take on a range of values from 0 to some number. So it's not going to be either of these range options. It's just going to be this first one here for I in my array. And if we were to write this, the loop would work such that I would be first 5 and then 6 and then 7 and so on. What is the appropriate way to make an iterator I that with every iteration becomes the number of iterations that have elapsed until as many iterations as there are elements in the array? So a similar sounding problem, and we have even the same array as the last one. But in this case, we want to know how many iterations have elapsed with each loop. So that's going to have no relationship to the numbers that are actually stored in this array. So in this case, we're going to want to use one of these range generators. So if we look at the second option here for I in range 0 to the length of my array, this is going to generate numbers 0, 1, 2, 3, 4, and so on up to, but not including the size of this array. So this is the option we want here, the second one. What is the appropriate way to make an iterator I that with every iteration can be used to retrieve the value at corresponding position of each element in the array? So a bit of a trick question. It's actually asking for the same thing as the last one. So if we want to be able to retrieve the value at the position corresponding to the iteration we're on, we simply need to count how many iterations we've been through and then use that count as our position. So we can use option B again here to do that. It's exactly the same as in the last question. So let's talk about DNA for a sec. And so in this series, we're thinking about certain types of data a lot. So we're thinking about sequences, matrices, and trees are going to be really common. And all of these are kind of representations or abstractions. And I think one of the first things to do is to think about the fact that these abstractions are representing, in many cases, real physical things that are, in actuality, pretty messy. And it's actually quite extraordinary that in the case of DNA, for example, that we have this abstraction that it is so robust. And this is a picture, an AFM image of a piece of DNA, and you can see that it's not linear in the same way that we have a typed series of A, T, G, and C bases. It's really a physical thing. And even at this level, the chemistry level, we're still relying on abstractions here. But in bioinformatics, we're increasingly or primarily dealing with this sequence object. And it's actually incredibly convenient, right? Because we can use the same tools that we use for processing computational data, classical informatics or classical computing, dealing with strings. We can use this to do biology. So we're going to talk about comparing two sequences, and some reasons for why you might want to do this. So one example is if you want to determine if two genes are of common descent, you might be comparing two sequences. Another possibility is inferring structure. So a lot of the structural biology is determining the structure not just based on imaging data or crystal structures, but based on the sequence itself. And because of the relationship between structure and function, this means we can also get information about function from the sequences. And what kind of sequences are we talking about? Well, we already know basically that it's going to be DNA, RNA, and protein. But a lot of these tools come from processing languages and processing cryptographic codes. A lot of classical computing tools we'll be using. What kind of differences might we expect when comparing two sequences? So of course, mutation. So if we're looking at genes that are maybe similar to each other but not identical, we might be looking for something like mutations. Insertions and deletions. So there are another type of mutation or difference between two potentially related or similar sequences. Fusions of sequences, duplications, transposons, this kind of thing. These are some examples. Here we're looking at two sequences. And here's a question. Are they different? It's pretty hard to tell. To what extent are they different? These are two different things we're asking. So we could, for example, just copy and paste this into a Word document and do a control F and maybe check that way to see if the sequences are identical. But as soon as we determine that maybe they're not identical, then we have a somewhat more advanced problem, which is asking to what extent are they different? And it turns out these two sequences are slightly different, but yeah, it is quite tricky to see exactly in what way. So there's a number of tools at our disposal, which we'll go through. Dot plots, score matrices, and then alignment techniques, global and local alignment techniques. So in the next few videos, we'll talk about these. But first, we'll start with dot plots. So this is a technique for visualizing two sequences. And it works like this. So you just place one sequence on one axis and the other sequence on, on another axis. And you have this matrix that's created as a result. And in the simplest case, we add a dot where there's a match. So here we have a D and here we have a D. So we fill in this square here. And this is called a dot, I guess. It's not really a dot, but here we have E and E are in agreement. So we add a dot here as well. But if we look, for example, E and D here are not in agreement, they're not the same. So nothing gets filled in here. So you do this for everything. If you were to compare the sequence with itself, if we were to look at DEE versus DEE, we would have this diagonal, right? That would be going across. These sequences are not identical. They're not. This is not a self comparison here. So we have this this mismatch on the end here. Here's another challenge. So here's linear inspection. It's quite tricky. This is not quite as bad as the first two sequences I showed you, but it is still quite difficult to just by eye know if these sequences are the same or not. But we can see here, okay, here's one mismatch. But humans are quite bad at this. What we are good at, though, is looking at images. And dot plots are a way to produce a kind of image, a matrix, but it is more visual, and you get some information, some structure from it. But what does this all mean? So this is pretty busy here. And of course, for DNA, we have the problem that A, T, G, and C, it's only 4 letters. So of course, A is going to occur multiple times. So we're going to get A's that align with A's in multiple positions, even if they're not necessarily, even if this is not necessarily telling us something about the structure. So a solution to this is to use what's called the sliding window method. And the idea behind this is to reduce some of this noise. And it's especially important for DNA. But it can be used for amino acid sequences as well. And the way it works is we have a window here. So it becomes a bit stricter, and we look to see if there is a perfect match within this window of everything within the window. And if that's the case, then we would add a dot for that particular position. And in this case, there's a mismatch, so there wouldn't be a dot there. And then you slide that window up. So here, this window is equal to 3, size of 3. We slide it up, and there's still a mismatch. We slide it up again. So now we're looking at the orange box here. Still a mismatch. This guy here, this Q and the K. And then we slide it up one last time to this yellow box here. And now we have a match between everything that's inside the window. So instead of looking simply one base at a time, we'd be looking at combinations. We'd be looking at, say, this block of 2 D and E at the same time. So the result of this is kind of filtering. And you can see for a window size 2, for those the same sequences that we were looking at here, we eliminate some of the noise, and some of the patterns start to become more evident. So here we can see something interesting going on. And sure enough, if we take a look here, we have a kind of pattern, right? It's AG, 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 AG. And we're getting a hit here. Of course, we got the same hit here, but it was kind of disguised by this noise. But what's happening is that AG here aligns with AG here as well, and AG here as well. So this 2 base repeat, especially for window size 2, it's having multiple hits, multiple 2 base hits along the sequence, and that's how we're getting this checkerboard pattern. Also, we see some patterns here as well, right? These lines, and if we look here, there's another kind of repeating motif here. We see GCTA, GCTA is occurring twice here. If we look further down, I think we have here we have it again, GCTA, and so you can see there's another diagonal slash here. That's basically these rows are agreeing with, of course, they're agreeing with across the sequence to their corresponding ones over here, but then they're also agreeing with this GCTA is also lining up with this GCTA further down here, and then again the other one that was located here in the middle. If we increase the window size even more, we get even more noise reduction, but we also start to eat away at the edges of our little checkerboard pattern here because we're approaching the edges. If it's a 3 base window, then we'll be encountering the edge of this pattern all the sooner. And you can see for window size 5, even more so. You can see patterns for, if we search for a small motif, for example, here we will look for this GATC, GATCA motif here along this sequence, which has it occurring multiple times, especially towards the end. We can see it happening a lot. GATC, GATC, yeah, GATC, I think here. So another kind of variation on this is a version where you tolerate mismatch. So in the strictest case, we go all the way back, if there's a single mismatch, then we would not put a dot. But another version of this is, rather than it be an all or nothing thing, we can simply count the number of mismatches and then scale that as a fraction of the total window size. So the best case scenario would be you have 8 out of 8 matches, but you could have, say, 6 out of 8, and then this would be a lesser value. And so here you can see, in this case, this sequence is being simply compared with itself. So we're looking kind of for intra sequence structure in this case. You can see some kind of noisy patterns here, and it's no longer this all or nothing situation. And dot plots, you can start to recognize certain patterns, certain structures, and it's good to know them. So here we have a kind of collection of different phenomena that you might encounter. So the first one is identity. So if we have our sequence 1 on this axis and sequence 2 on this vertical axis, of course, if you get this strong diagonal going down the middle, then that means you have dots or matches at each position in both sequences corresponding to each other. Duplication events, so that you'll see that whenever there's a parallel line. So we have a diagonal, the two sequences are in agreement with each other, but they're also in agreement with themselves later on. So this, if we were to pick like a row here, it's got a dot here, but then also a dot later down. Palindrome, right? A palindrome is something that you can read forward and read backwards as well. So we have a match not just along the main diagonal, but orthogonal to that diagonal as well. D is similar, just a partial palindrome. So here we have, the entire sequence is not palindromic in this case. In the case of E, we have similar to the checkerboard pattern that we saw with the GA, 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 if you recall. So this is a microsatellite repeat, and you would see these kind of boxes whenever that's happening, like a die-based repeat that's happening over and over again. And a kind of a similar version of this, mini satellite repeats, maybe a sequence of 6 bases or 10 bases that's occurring over and over again. Homology, or partial match. So here we have two sequences that have a strong diagonal, but not a perfect diagonal. So there's some gaps in here. And this you might see when the sequences are related to each other, but perhaps not identical. And then finally, the last case, a deletion and or insertion event. So you can see here that in sequence 2, the vertical one, we're adding some stuff. So that's an insertion event. But this is also the same as saying we deleted something from sequence 1 on this axis. Okay, so here's an interesting one. What could we be looking at here? So I'll tell you right now that this sequence is being compared with itself. So this is another case where we're looking at internal structure or self structure. So what is it? It's in fact a song. So this is Somewhere Over the Rainbow by Judy Garland. And you can see the lyrics here. And I think this is an interesting analogy because songs tend to have some structure to them. And you can see, you can use the exact same tool to analyze the structure of a song that's been converted into a string of text. You can use the same tool to look at DNA sequences or protein sequences, and you see some of the same motifs that we talked about. Here's a duplication event. So here, that's this first lyric here, Somewhere Over the Rainbow, is being repeated. And you can see that it's occurring not just at the beginning, but also later on and towards the end a few times. Here's another example of a song. I think I chose this one because it's got some interesting structures in it. So this is DNA by Kendrick Lamar. And you can see we have actually these mini satellite repeats or microsatellite repeats. And of course, that is this lyric here. I got, I got, I got, I got. So here we get this kind of wavy checkerboard pattern, and you can see it occurring in a bunch of places throughout the song here as well. And here I've tried to highlight in orange all the places it occurs. Also, these stretches here, so duplication events, repetition, basically. So the green here is this line inside my DNA, which he mentions quite a few times in his song. And then smaller versions of this, you can see some, so it's decaying a bit at the end, and then he restores the full lyric here, adding back this word inside. So you see a different signal there. Also, here in the song, we've got some other repeat events. And here's some other songs. So yeah, you can see a lot of patterns here, and it tells you a lot about the song, right? You can, in a glance, kind of see the structure of it. You can see here in this song, we have a kind of intro chorus, this block, and then you see the same thing occur twice towards the end. And from music theory, this is like the coda, right? It's some theme that you saw earlier in the song, and that's repeated a couple times at the end of the song. You can see also some interesting structures here, some repetition, something like a mini satellite repeat here also occurring. So yeah, that's an analogy and how we can think about structures of sequences. Dot plots, there's some tools out there to make them. So if you just want to use an online tool, there are things like dot plotter and dot matcher, but also Python and R packages and some downloadable options. And in the exercises, we'll be playing with these as well. Okay, and I think that's it for this video. In the next video, I'll be talking about score matrices and quantifying similarity. So thank you and see you there.